Welcome to BioMed Research Digest 2026-0602, today's 12 stories. Story 1, this new drug could change how we treat one of the deadliest cancers, Daraxonrasib for pancreatic cancer. Story 2, FDA approves Xocova and ensitrelvir, the first oral option to help prevent COVID-19 following exposure. 3, Izalontamab Brengitecan Iza-Bren demonstrates significant survival improvements in triple-negative breast cancer and esophageal cancer. Story 4, high-precision base editing therapy CS206 demonstrates durable VOC reduction in sickle cell disease. Story 5, cancer jab Amivantamab can eradicate entire tumors in patients, trial shows. Story 6, FDA accepts new drug application for Roche's Giredestrant in ER-positive early breast cancer. Story 7, European Commission approves Amgen's IMDYLLTRA for extensive-stage small cell lung cancer. Story 8, CRISPR-Screen informed engineered T-cell therapies, frontiers in immunology. Story 9, Wockhardt's Zaynich makes history as India's first fully homegrown FDA-approved drug. Story 10, rational design of immune gene therapy combinations via in vivo CRISPR activation screen of tumor microenvironment modulators, cancer discovery, AACR. Story 11, unveiling the shared gray matter signature between Alzheimer's and Parkinson's disease. Story 12, Site 4 drug, predicting drug-binding target sites with an AI agent. In a landmark study published in Cancer Discovery, the high-impact journal of the American Association for Cancer Research, scientists have used a powerful genomic tool called CRISPR activation to systematically identify which factors in the tumor microenvironment can be leveraged to boost anti-tumor immunity. This work, released in June 2026, marks a meaningful shift in how CRISPR technology is being applied in oncology. Most people associate CRISPR with gene editing, cutting, and correcting DNA. But this study uses a variation called CRISPR, which activates genes without permanently altering the DNA sequence. The researchers conducted this screen not in a petri dish but in living animal models, making the findings biologically richer and more relevant to what actually happens inside a tumor. By systematically switching on hundreds of candidate genes and observing which ones tipped the immune balance toward attacking the tumor, the team built a prioritized map of actionable targets in the tumor microenvironment. Why does this matter? Because the tumor microenvironment, the complex ecosystem of immune cells, blood vessels, structural proteins, and signaling molecules surrounding a tumor, is one of the biggest reasons immunotherapy fails in solid cancers. Tumors actively suppress immune responses, and identifying which factors, when activated, can reverse that suppression is enormously valuable. Until now, finding the right combinations of immune therapies has largely been a process of trial and error. This study offers a rational, data-driven alternative. The implications are far-reaching. For pharmaceutical companies investing in immune gene therapies, this kind of systematic screen could dramatically reduce the time and cost of identifying effective combination strategies. For patients with solid tumors who have not responded to existing immunotherapies, it opens a path toward more precisely targeted treatment regimens. There are caveats to keep in mind. The study is preclinical, and the gap between mouse models and human biology remains real. CRISPR tools can also produce off-target effects that may complicate interpretation, but as a proof of concept that CRISPR can be repurposed as a discovery engine for oncology combination therapy design, this research is a compelling step forward, and one the field is likely to build on significantly in the years ahead. A new neuroimaging study submitted to Archive in June 2026 has produced what researchers describe as the first vertex-level quantification of shared gray matter changes between Alzheimer's disease and Parkinson's disease. The work comes from an international team spanning the University of Queensland, QUT, and the ARAMIS Research Group in France, and it uses a novel statistical framework to ask a fundamental question. Do these two devastating neurodegenerative diseases share a common footprint in the brain? The answer, according to the data, is yes, partially. The team found a significant positive correlation between the gray matter patterns of Alzheimer's and Parkinson's patients, with a brain-wide gray matter correlation coefficient of 0.24. That might sound modest, but when you consider that millions of people worldwide are living with one or both of these diseases, even a modest shared biological signature could unlock meaningful new approaches to early detection and treatment. Drilling deeper, the researchers identified 9 specific clusters in the brain, comprising 106 individual measurement points, where the overlap was most pronounced. Particularly notable were the bilateral putamen and the right nucleus accumbens, structures deep in the brain involved in motor control and reward processing, both of which showed reduced thickness associated with both diseases. Crucially, these findings held up across different stages of each disease and were independently replicated using data from the UK Biobank, one of the world's largest biomedical databases. Why does this matter? Currently, screening and monitoring for Alzheimer's and Parkinson's are treated as entirely separate clinical enterprises. If shared neuroanatomical markers can be identified early, before symptoms become severe, it may be possible to develop combined screening tools or to identify patients at risk for both conditions simultaneously. Shared pathological regions like the putamen might also become targets for interventions designed to slow neurodegeneration across disease boundaries. There are important caveats. This is a preprint, meaning it has not yet been peer-reviewed. The correlation, while real, is modest and the causal story is not yet clear. But as a methodological contribution and a starting point for cross-disease research, this study represents a genuinely novel contribution to our understanding of neurodegeneration. A new AI tool called Site4Drug, accepted to the ICML 2026 workshop on generative and agentic AI for biology, takes on one of the most underappreciated challenges in pharmaceutical research: figuring out exactly where on a protein a drug should bind. Developed by Tihan Kim and colleagues, the system functions as an intelligent agent that doesn't just predict binding pockets. It reasons through biological constraints to produce ranked, evidence-backed recommendations for targetable sites. What makes Site4Drug distinctive is its focus on membrane proteins, the class of proteins that stud cell surfaces and act as gatekeepers for signaling and transport. These proteins, including receptors, ion channels, and transporters, account for roughly 60% of all drug targets, yet they remain poorly served by existing computational tools, which tend to be designed for soluble proteins with well-defined pockets. Membrane proteins are harder to analyze because their binding sites may be buried in or near the lipid bilayer, and they are heavily modified by post-translational processes that can hide or expose key regions. Site4Drug incorporates all of this into its analysis. Protein topology, hydropathy, disulfide bonds, post-translational modification propensity, domain context, and sequence information are all factored into its site-finding process. Crucially, the system doesn't require users to know upfront whether they want a small molecule, an antibody, or a peptide-based drug. It can recommend the appropriate drug modality based on the same evidence it uses to identify the site. This is important because a site that looks chemically attractive for a small molecule might be sterically blocked in practice, and vice versa. Another standout feature is the system's traceable decision log. Rather than delivering an opaque prediction, Site4Drug documents the reasoning behind each recommendation, including risk flags and evidence summaries. As AI tools become more embedded in drug discovery pipelines, the ability to audit and explain AI decisions will be essential for both scientific credibility and regulatory compliance. That said, Site4Drug is a workshop paper, which means the results are preliminary and have not been subjected to full peer review or rigorous benchmarking against competing tools. The potential is clear, but the field will be watching for comprehensive validation studies before this moves into mainstream drug discovery workflows. CS206, a novel base editing gene therapy for sickle cell disease (SCD), has demonstrated durable suppression of red blood cell sickling and significant reductions in vaso-occlusive crises (VOC) and hemolysis. Unlike CRISPR-based approaches, CS206 uses precise single-base correction without introducing DNA double-strand breaks, potentially offering a safer profile. This positions it as an important advancement over existing gene editing therapies for SCD. A clinical trial has shown that Amivantamab, a bispecific antibody delivered as a subcutaneous injection, can eradicate entire tumors in some patients, offering a major advance over intravenous treatment regimens. The subcutaneous formulation significantly improves patient convenience and accessibility of treatment in outpatient settings. This represents a meaningful shift in how bispecific antibody therapies can be delivered in oncology. The FDA has accepted Roche's new drug application, NDA, for Giredestrant, a next-generation oral selective estrogen receptor degrader (SERD) for early ER-positive breast cancer. Giredestrant is described as the first major endocrine therapy advance in early breast cancer in years, with an FDA decision expected by November 30, 2026. This could establish a new standard of care for one of the most common cancer types. The European Commission has approved Amgen's IMDYLLTRA tarlatamab, a DLL3 targeting bispecific T-cell engager for extensive-stage small cell lung cancer (ISCLC) based on phase 3 to LL5304 trial data. ISCLC is an aggressive malignancy with very limited treatment options and poor prognosis. This approval marks a significant step forward for immunotherapy in this historically underserved cancer type. A peer-reviewed paper in Frontiers in Immunology describes how CRISPR screens are being used to identify and validate targets for next-generation engineered T-cell therapies, including CAR-T approaches. The work bridges target discovery and clinical translation, with several trials underway to validate CRISPR-Screen identified candidates. Safety considerations around genomic integrity and off-tumor toxicity are also systematically addressed. Zaynich WCK5222, developed by India's Wockhardt, has received FDA approval, becoming India's first fully homegrown FDA-approved drug. The novel antibiotic demonstrated high efficacy against difficult-to-treat urinary tract infections in the global Phase 3, Enhance-1 trial outperforming meropenem. This is significant for the global fight against antibiotic-resistant bacteria. Published in Cancer Discovery, this study uses an in vivo CRISPR activation, CRISPR-Screen targeting the tumor microenvironment, TME, to identify factors that promote anti-tumor immunity. The findings enable rationally designed immune gene therapy combinations with improved precision. This approach moves CRISPR applications from gene correction towards systematic drug combination design in oncology. This preprint presents the first quantification of shared vertex-level gray matter changes between Alzheimer's disease, AD, and Parkinson's disease, PD, using high-resolution MRI data aggregated across large datasets. Using a novel statistical framework, SUM-R2 regression, researchers identified overlapping neuroanatomical signatures that could inform shared pathological mechanisms and biomarker development for both diseases. Site4 drug is a new AI agent that identifies and ranks druggable binding sites on proteins, particularly addressing the challenge of membrane proteins where accessibility and post-translational modifications complicate site selection. The tool outputs ranked targetable regions with explicit constraints, evidence summaries, and reasoning, improving early-stage drug discovery decision-making. This represents an advance in AI-assisted drug design pipelines.